Okay, now we're going to talk about transmission of information by neurons. So, neurons function by being able to undergo rapid changes in the electrical potential across the cell membrane. So, the electrical potential is the distribution of ions, which creates a difference in the electrical charge on each side of the membrane. So, that's pretty important, and we'll talk a little bit about that. So, it's really key to the neuron's function. If you have a different electrical charge on each side of the membrane and it's closed, which it is at its resting state, then you have the potential for an electrochemical gradient. And so once you open up gates into the membrane, that charge can move from one side of the membrane to the other, and it can change the overall state of the membrane itself. So the resting state is called the resting membrane potential. That is when the neuron is not excited, the neuron is just sitting there. It's waiting for its next impulse. So it's the difference in electrical potential across the membrane when the neuron is not transmitting anything. It's resting. Okay, so the cell membrane itself, it allows for the separation and storage of electrical charge. So it's like a storage battery, sort of. So the typical resting membrane potential is minus 70 millivolts. So there's a concentration gradient. There's more sodium on the outside of the cell. There's more potassium on the inside of the cell. There are these big fat anions, which are negative ions trapped inside the cell. They're kind of neutralizers. And then we have big fat chloride ions on the outside of the cell as well. And the chloride and the anions don't move back and forth through the channels. The channels are specific for sodium and potassium, but we'll talk more specifically about channels in a minute. The difference in the positive and negative charge inside and outside the membrane control that ion movement. So those big negative anions are trapped inside the cell because they're big and they just don't fit through the gates. We have a pump that is run by ATP, the energy currency of our cell, all our cells. And it actively carries 2 potassium and 3 sodium out of the cell with each cycle. It uses 1 molecule of ATP. So it maintains that unequal distribution of potassium and sodium, more sodium outside the cell, more potassium inside the cell, and that's what creates that electrical chemical gradient. And that is setting you up for being able to move charge across the membrane once it opens up. So this picture shows your minus 70 millivolt charge. The inside of the cell is the blue part. We have anions and potassium. The outside of the cell is the extracellular space. That's much more positive. We have more sodium out there than we have potassium on the inside. So the inside of the cell is relatively negative. That's that negative 70 millivolts. The outside of the cell is relatively positive. So we have different, okay, I already talked about this, the pump and the negatively charged molecules trapped inside due to their size, and passive ion channels. So let's talk a little bit about channels. Okay, so there are 4 different types of channels in the membrane, and they allow ions to flow across the membrane. The first type is called a leak channel, and it's just like it sounds. It's a little hole. It's always open. Sodium can leak through little by little. It's sort of like the hose in a drip irrigation system. There's not a lot of water coming out, but there's a little coming out little by little, and so sodium can follow its electrochemical gradient and go into the cell if it wants to through those channels. But just like the drip irrigation hose, not a lot is going through at one time. It's like one little sodium molecule at a time. It's just barely leaking through. The other 3 channels are gated membrane channels. So when what they mean by gated is those channels only open in response to a certain stimulus, and they close when the stimulus is removed. So there are 3 different types of gated channels, and you should know those 3 different types of gated channels and what the stimulus is that opens the individual gated channels. So we talked about the leak channels. A small number of ions leak at a slow, continuous rate. That's our drip irrigation. Our gated membrane channels, the 3 of them, are modality-gated channels, ligand-gated channels, and voltage-gated channels. So a modality-gated channel opens in response to a mechanical force, temperature change, or a chemical change. So if you think about modalities, the things we did last quarter in 111, soft tissue mobilization, temperature, hot-cold treatments, ultrasound is a mechanical force, iontophoresis, chemicals are changing the cell membrane, but also that's just any pressure on a sensory nerve ending, any temperature change, chemical change, or mechanical change that stimulates that sensory nerve ending, that opens those modality-gated channels, and it sets off the cascade of nerve transmission. So we'll look at another diagram from the book in a few minutes that shows the whole cascade. Okay, so ligand-gated channels open in response to a neurotransmitter binding to the surface of the channel receptor on a postsynaptic cell membrane. So ligand means bind. And so in order to open that channel, something has to bind to it. And it's a specific thing that's bind to it. Each channel is a protein that only a specific other protein can bind to activate it and open it and let those sodium ions in. So all of our neurotransmitter receptors, dopamine receptors, acetylcholine receptors at the neuromuscular junction, which we'll talk about in detail a little bit later, those are all ligand-gated channels. They're on the postsynaptic cell membrane. So they're somewhere on a dendrite or on the soma or on another axon, and they are, when the presynaptic terminal of one axon releases neurotransmitter into the synapse, that neurotransmitter binds to those channels and it opens them so sodium can get through and change the electrical potential of the cell. Okay, the third kind are voltage-gated channels. They open in response to changes of the electrical potential across the cell membrane. So you can kind of see a little chain effect happening here. We will talk about the whole chain effect in a minute, but you'd say, hey, a modality-gated channel opens in response to a mechanical force. That opens the channel, it changes the electrical potential across the cell membrane, and hey, maybe it opens some voltage-gated channels, and the electrical signal propagates down the neuron and goes to the presynaptic terminal and releases neurotransmitters, and then those neurotransmitters bind to a ligand-gated channel and they go on down the chain. So these are all closely related, but they all only respond to a specific stimulus. And once the stimulus is gone, the channels are closed. So mechanical forces, the mechanical force is taken away, those modality-gated channels close, and the cell returns to its resting state. The ligand-gated channels, when the neurotransmitter leaves, gets you get a neurotransmitter gets reuptaken into the presynaptic terminal, the channel closes because it doesn't have its stimulus anymore. With the voltage-gated channel, the electrical potential changes and then it goes back to its resting state and the voltage channel closes. So once you remove the stimulus, the channel closes. Okay? So this is a figure 2.3 A and B from the sodium ion channel. This is a voltage-gated channel. You can see the extracellular fluid is more positive than the inside of the cell, which is more negative. When the potential changes, you see on figure B, the outside is more negative, the inside becomes more positive, the channel opens and ions can go through. So that's its little reason for living. So electrical potential, it's a difference in electrical charge, which is carried by ions. So how many ions you have outside the cell versus inside the cell, that's what determines your electrical potential. A rapid change in the electrical charge across the cell membrane transmits information across the length of the along the length of the axon. So you can, it's the same thing when it gets to the end of the axon at the neuromuscular junction. When it gets to another neuron, you can release those chemical transmitters to excite that neuron, but you can also release chemical transmitters, acetylcholine specifically at the neuromuscular junction, to change the potential, the electrically excitable membrane of the muscle. So that electrical potential coming down the neuron, it's just going its own little way until it reaches its target cell, and then you know what it's going to do. So the neurons in between, they're just transmitting it. They're saying, okay, I'm passing this along the line to the next guy, until you get to the end of the chain, and then something happens. So the end of the chain might be another neuron, it might be a muscle, it might be an endocrine gland that's going to release some hormones. Kind of cool stuff. So I think I'm going to wrap this up, and we will then resume in another recording to start talking about electrical potentials. Let's see. Okay, so this, yep, we'll talk about electrical potentials in the next one.